All right, so now let's talk about DNA. Here you have a few different visual representations of DNA. And so things that words that are used to describe DNA are double helix. And when you're talking about a shape that's helical, you're talking about something that is kind of spiral shaped. And it's a double helix because there are two of those spirals going side by side. So a lot of times DNA is kind of compared to a ladder, and that's kind of what it looks like. And we'll kind of dive into details about what the sides of that ladder would be and what the steps of that ladder would be. And so first, let's talk about Rosalind Franklin. Rosalind Franklin used this X-ray diffraction patterns, and so you'll see that down here and actually up here too, to discover that DNA twisted around each other like a double helix, right? So she is the one who first saw this in her X-ray diffraction, and the angle of the X pattern suggested that there were two strands. Right, so she was able to tell a lot more from this than I can tell. But the unfortunate thing, though, is that she didn't really get the credit that she deserved for this. The people that got all the credit for this surprise were men, right? And so Watson and Crick, and let me actually move this out of the way right here. And so Watson and Crick uh, while trying to build a 3D model of DNA, Watson was shown Franklin's photograph and within weeks discovered out the structure of DNA. So there you go, right? They very luckily were able to see the work that Rosalind Franklin was doing, and they copied that work, or they took that work, and they were able to construct in only 2 weeks the model in 1953. So it looks like a spiral staircase. I like this GIF right here because it kind of animates that it does kind of look like a spiral staircase or a spiral ladder, right? So that is the double helical shape of a DNA, right? It's two strands and then they spin around each other kind of like a spiral. And so DNA, if you ever want to impress anyone, let them know what DNA stands for. Not a lot of people do, right? And that is deoxyribonucleic acid. And so deoxyribonucleic acid, for short, we just call it DNA, right? But DNA is a molecule of life, as it says right here. Remember, y'all, that DNA is a genetic code, and the genetic code in some parts you see here's a cell, and then this blue part is a nucleus. Remember, the DNA lives within the nucleus in our cells, eukaryotic cells, and usually it just kind of looks like thread or just like a bunch of spaghetti spooled out there. But before these cells divide, they actually thicken up into these chromosomes right here, and we'll talk about those in the next lesson in more detail. These chromosomes right here, though, they need to thicken up because eventually what's going to happen, let me just kind of draw this little thing right here. So you can see eventually these chromosomes will just kind of get split right in half. I'm trying to make these little dash lines. So a chromosome will get split right there, and half will go this way into one cell, and this half will go this way into another cell. That comes later, but I just kind of wanted to let you know why is it that it condenses into this chromosome, and it's because it's eventually going to divide into two different cells. Right, but here you have this DNA kind of like that thread, a spaghetti looking thing, and here you see the double helix shape. But something to remember, y'all, is when we talk about genes, a gene is just a small segment of DNA, not necessarily like small. I guess it's relative, right? It's a piece of DNA that codes for a trait, right? So, like this gene right here could be part of a gene for like eye color or hair color, or whether your hair is straight or curly. But again, it's just a piece of DNA that codes for a protein or a trait, right? And so, in this case, that's what a gene is. So the function of nucleic acid. So we talked a little bit about the structure, right? It's this double helical shape, spirals around each other. And so that is the structure. And we'll talk a little bit more about the structure. But the function, the thing that it does that is so important to us is it stores and carries genetic information, right? So those A's, T's, C's, and G's that we talked about, and we'll talk about in a little bit more detail, those are the four letters that make up that code, right? And this code is passed from parent to offspring. And here there's this gif of Ice Cube and his son, and they look very similar, right? There are some differences. One's a little bit lighter than the other, and their eyes are a little bit different. But for the most part, if you look at them, especially their faces, they look very similar, right? And that reason why is because this his son right here, half of his DNA came from his dad, Ice Cube, and the other half from his mom. Right. And so he looks like his parents, as a lot of you probably do. If not, then great your grandparents or your great grandparents, right? And so it is important to remember that sometimes you're a mixture of both, right? And so you really don't look too much like one or the other because you're just almost an even mixture of the two parents, right? So again, we talked a little bit about this, but let's look at this in more detail. Right here, this is the structure of nucleic acids. So we talked about this when we talked about biomolecules, right? The nucleic acids are made up of building blocks called monomers. But if you're talking about the specific monomer for DNA, it's a nucleotide, right? This nucleotide. And this nucleotide is made up, and it's important to know this, is made up of a phosphate. You see the phosphate right here. It's made up, and let me get this guy right here. So the phosphate is right here. You see the sugar is right here. And here you have the base. Remember, y'all, when you see the base, remember these letters right here, right? A, T, C, and G. And if we're kind of breaking that, or actually elaborating on that, A is for adenine. And let me start up here, right? T is for thymine, right? So whenever you're talking about this T right here or this T right here, you're talking about thiamine right there. Cytosine right here is this C. So if you're talking about cytosine, C you see is right here. Well, not the G, sorry. C is right here, C is right here, C is right here, but it's also right here, right? That is cytosine. If you're talking about A, adenine, you'll see it right here. A for adenine, A for adenine. And so for G, guanine, you see that guanine right there? That is this G right here, and then this G here, this G here, and this G here, right? That is guanine. So what's important to remember, y'all, is this, and we'll see this in a little bit too. When you have one of these little building blocks right here, it's the phosphate, the sugar, and the base. That is a monomer, right? And here you see, I can just, let me circle just one of these monomers right here. So this would be a monomer. This would be a monomer. This would be a monomer. But when you get a whole bunch of these monomers together, it's what you call a polymer, right? So polymers, like the generic name, is what you call a bunch of nucleotides put together, but there's specific names for DNA. There's another one for RNA, and there's another one for ATP, and some others. But that's the main focus that you'll probably be familiar with from this class, right? So again, the monomers are nucleotides, and they are made up of a phosphate, a sugar, and a base. The sugar and the phosphate, this is what you call the sugar phosphate backbone. And we'll see this in more detail. But this right here is a sugar phosphate backbone. Let me kind of highlight it there. And then when you're talking about the nitrogen base, these are the nitrogen bases right here. This right here, this right here, this right here. All right. And so again, base pairing, or not again, because I haven't mentioned this before, but one of the things that you need to remember too is base pairing, which means whenever you have an adenine like you do right here, that will always be matched up with a thymine, right? So A's and T's are always matched up together. They complement each other. So AT, TA, and it doesn't matter which order, TA, as long as there's an A, it will be attached to a T. Here, if there's ever a C, it will be attached to a G, or a G to a C, right? It doesn't matter which order, but C's and G's always go together. Right here, and I think someone said once that it helps them remember when they think of apple trees and car garage. And so apple trees, car garage. And so, yeah, whenever you have an A, it'll be matched with a T. Whenever you have a C, it'll be matched with a G, right? And that is base pairing. And so a little bit more detail on these nucleotides. Remember, again, there will be some repetition, y'all, but it's to your benefit, right? So it has three parts, the nucleotide. And remember, nucleotide is a monomer of a nucleic acid, which means that it is one of the little links that make up a big chain of like DNA or RNA. And so these are the three parts of the nucleotide: the phosphate group, and here you see the phosphate, the pentose sugar. And pentose, when you think of the word penta, it means five, right? The pentagon has five sides, a pentagram has 5 points. Same thing here with this pentose sugar: one, two, three, four, five. Right, so it is a pentose sugar. And remember, y'all, what we've talked about before. If you see this three letters, OSE, it means it's a sugar. Right, and so then you have the nitrogen base. Whenever you're thinking of the nitrogen bases, those are the four letters that we talk about, right? A, T, C, and G. Right? So this is what makes up the nucleotide, the three parts: the phosphate group, the pentose sugar, which is ribose, and then the nitrogenous bases or nitrogen bases, which are adenine, thymine, guanine, cytosine, or A, T, G, and C. Right, and so here you have the sugars, ribose and deoxyribose. And so whenever you're talking about DNA, remember it's deoxyribonucleic acid. When you're talking about RNA, we'll learn about that in a little bit, but that is a ribose, right? That is where the RNA comes from. So right here, this would be, just draw that up, RNA. And then on this side, this would be your DNA. Right? So these are the sugars, right? And the pentose sugars, remember, 5 sides. And then the nitrogenous base, again, these are adenine A, guanine G, cytosine C, and thymine T. And here you see it broken down, or in a little bit more detail, right? You have the sugar phosphate backbone, right? So these guys right here are the sugar phosphate backbone. And then these guys right here, let me put them in a different color, are the nitrogen bases, or like it says right here, base pairs, because they're always paired up, right? A with T and G with C. Right, so remember, AT, adenine, thymine, GC, guanine, cytosine. And remember, it doesn't matter if the A is first or the T is first. It doesn't matter if the G is first or the C is first, right? A with T, C with G, and vice versa. What's really important to remember, and remember this because you could see this on an exam, is what kind of a bond holds the two strands of DNA together, right? And as you can see right here, the bond that does that is this hydrogen bond. But remember this, y'all, this hydrogen bond is a weak hydrogen bond. So I'm going to put this right here next to that. This hydrogen bond, and not very good. I'll just put it right here. So this hydrogen bond right here is a weak hydrogen bond. And that's important because this DNA molecule right here has to eventually split right down the middle. And so I'm just going to draw this little dash showing where it's going to split, and half is going to go this way and half is going to go this way. And so it's important that this is not very strong. It's strong enough to hold it together, but it can be pulled apart if it has to, which eventually it does have to. Because remember, y'all, cells divide, right? And if it's going to divide, it's not like one cell can just keep all the DNA for itself. The cell has to actually double up on its DNA and then separate it into different cells, right? So that's why this hydrogen bond is important, that it is weak. So base pair rules. I've said this a few times, but again, it's all about that repetition, right? Adenine and thymine. So wherever you see an A, there is going to be a T, right? Adenine, thymine. Wherever you see this G, there is going to be a C. So guanine and cytosine, right? Or right here, guanine, cytosine, thymine, adenine. And then here you see the sugar and the phosphate. Remember, this sugar looks like a little house, right, with 5 sides, the pentose sugar. And then this P right here is a phosphate. So again, these are the sugar phosphate backbones, and these are the nitrogen base pairs, right? Nitrogen base pairs, all of them. Right. And so the sides of the DNA are the sugar phosphate, right? These sugar phosphate backbones right here. And then the base pairs are held together by this hydrogen bond. And here you can see that. Let me erase this right here. Here you see those hydrogen bonds, right? Right there, holding it there, holding it there. And these are weak bonds. And that means that it can eventually separate right down the middle. Right. So base pairs, whenever you see, again, an A, that means you're going to have a T. Whenever you see a T, you're going to have an A. So go ahead and complete this right here. What are the complementary bases? And I'll go ahead and help with this first one right here. So here, remember, if you have an A, that means it is complemented by a T. If you have a T, that means it is complemented by an A. One. And then here, when you have an A, that is complemented by a T. Right. And so then just do the rest this way. All right, and again, you have the thiamine, adenine, adenine, thymine, guanine, cytosine, ATCG. And here you see and remember that part of it is that it is a weak hydrogen bond. So these hydrogen bonds, right? Just finished talking about it a second ago. Hydrogen bonds occur when hydrogen is attached to one element but is attracted to another nearby element, right? So that's what's happening here is it's attached to one but it's attracted to another element nearby, and that's kind of what keeps the two DNA strands together. Hydrogen bonds are weak, but there are millions of them in a single molecule of DNA. So even though they're weak, because there are so many of them, it holds them together. But again, it's a weak bond. So eventually, this DNA molecule right here will get separated into two. Right in here, it looks a little bit different, but here is a phosphate. Here is a five-sided sugar. Here is a T, a phosphate, sugar, cytosine, the C, phosphate, sugar, the G. Right. So again, these hydrogen molecules are going to split. Sorry, these bonds are going to split right down the middle. And then half of this DNA is going to go to one cell over here, and the other half is going to go to a cell over there. Right. So how the code works. The combination of A, T, G, and C determines what traits you might have. And for example, this gene right here, right? So let's pretend this is a gene right here. This gene CAT, that codes for proteins that would lead to purple hair, right? But if you had this gene right here, and let me just write that out really quick. So this would be an example of a gene. This one right here, down here, would be an example of a gene. And so again, gene is right a little piece of DNA that codes for a protein that leads to a trait like brown eyes, blue eyes, curly hair, straight hair. And so right here you have this gene TAC. And so this gene right here codes for orange hair in a cat. Right. So that is what all those combinations of A's, T's, G's, and C's, they lead to all of the diversity that you have on Earth. Right. Think of the bases of DNA like letters. Letters form words and words form sentences, and there are endless combinations. And this is what I was just saying, right? Think of all the diversity on the planet, all of these different organisms, from these microscopic ones to these up here, ladybugs, worms, mollusks, and even to humans, right? Everything that makes us different is just because we have combinations of these letters right here: A's, T's, C's, and G's.